Here is a 3D representation of the histology of the salivary gland. Let's see the structures one by one. In this model, I have represented only a small piece of the salivary gland because the file size was really huge. When I started to add more of this, it's like growing in gigabytes. So let's assume that we are looking at the corner of a salivary gland, and this is the entire outer surface. You can see that connective tissue fibers, which are highlighted here, cover the entire gland, and that is called the connective tissue capsule of the gland. Notice that the capsule sends connective tissue septa deep within the gland to divide the gland into several lobes and lobules, as I said. This region, for example, can be considered as a small lobule where you see the septa. And the septa and the capsule are actually keeping the gland in place. Plus, they are also helping the blood vessels that you see here or the ducts that you see here to go deep into the gland. The pink and blue structures that are visible here are nothing but the acini. So you can see that in 3D, the acini are more like balls. The pink ones are the serous acini, the blue ones are the mucous acini, but in a section, they are looking like this. The acini can be of any size. Usually, the serous acini are smaller than the mucous acini, and also within the same type of acini, you can see bigger acini and smaller ones. That's either because they are actually bigger or they are cut in a different plane. So compare this one. If you are going to make a section along this plane, then the acinus will look bigger in the section. But if you are going to make a section here, then the acinus will appear smaller. Let's go into the details of the serous acinus. What you see around the acinus is the basement membrane, and the individual cells are sitting on the basement membrane. And that is what I said in the lecture, that all glands are examples of epithelium. So as a rule, epithelium has got a basement membrane, and all the basal cells will be touching the basement membrane and they are packed together. The cell of a serous acinus is pyramidal in shape. Again, you can see that it's pyramidal in 3D and it's more triangular in a 2D section. The top of the cell is narrow, and that's pointing towards the lumen. So this is the lumen, and you can see that it's small, and that is nothing but the mouth of all these cells. If I isolate only this serous acinus here and remove the connective tissue around, you can see that in a cut section, the acinus looks like a cut orange. Now I'll remove the basement membrane, which is lining the entire acinus. You can see that the cells are again arranged same as orange fruit or something. And you can see that all the individual cells are pointing towards each other, and that small opening is the lumen of that particular acinus from where a small duct will arise. We can even explode this model to see what I'm talking about. Yep. So see here, all the individual cells were initially packed together. Now if I explode it, means if I remove all the cells apart, you can make out the acinus and how the individual cells are related to each other. The base of the cell is wider, and towards the base, we will see all the organelles that includes your nucleus, mitochondria, and rough endoplasmic reticulum, etc. The advantage of this model is that you can zoom to any level and see you are actually seeing the rough endoplasmic reticulum in 3D with the ribosomes sitting on it, giving them the rough surface. And you can also see the mitochondria if you want. You can zoom in to see the crista or the partitions within the mitochondria. The green colored granules represent the zymosin granules, which are nothing but the secretory precursors of serous saliva, and the granules are releasing saliva into the lumen here. you can see that even though the cells are tightly packed, at some places there is a loose space between the cells. So what does it mean? When the saliva is secreted from the cell, we want all the saliva to go into the lumen of the acinus and eventually go into the duct. We do not want that saliva to go back and leak between these cells. Okay, so how is that prevented? That's where the tight junctions are coming into place. If you look at the entire arrangement, the tight junctions are like staples. They staple the two surfaces of the cells within the acinus. Thereby it prevents any leakage of saliva from here going back between the cells. This junctional complex, which is nothing but the tight junctions holding all the cells together, can be seen only under electron micrograph details. These are not visualized under your light microscope. The other interesting feature of the acinus is, see here, if I go and zoom further, I can see the basement membrane and one of the cells here. Between the cells and the basement membrane, I can see another cell, and that's nothing but your myoepithelial cell. These myoepithelial cells, as I said, they surround the acinus. So you can see one here, maybe one in the other plane. They are modified smooth muscle cells. They will contract so that they can squeeze the acinus and throw the saliva out where needed. And they are placed inside your basement membrane. Now if I isolate those cells, you can see that they are actually star-shaped cells, okay? Whereas in the 2D images, we can always see them like a small line, but in reality, they have got lots of projections from them. For this reason, these are also called basket cells because they enclose the acinus like a basket. These projections are helping in squeezing the acini and thereby releasing the saliva from them. Again, another feature that can be appreciated only in 3D. If you go into the details of the mucous acinus, we will see that some features are common with the serous acinus. For example, the basement membrane, which covers the entire acinus, and the cells are sitting inside the basement membrane, touching it. And you can see that the shape of the cells here is columnar or cylindrical cells rather than pyramidal cells that we saw in the serous acinus. Let's compare the differences now. In the serous, the lumen is very small, whereas in the mucus, the lumen is big. The reason is the serous secretions are really watery, whereas the mucus secretions are big and thick. Okay, see, look at the size. These are the zymosin granules for the watery secretion, whereas these are the mucin droplets or mucus. So to have big mucus secretion, we will need a bigger lumen. The other common details are within two cells. For example, we have got the tight junctions, which are the junctional complexes keeping the mucus in place and not allowing it to leak back between the cells. It is because of that mucus, you can see that the organelles within the cell are completely pushed down to the basal surface. So you can see that the nucleus is really squashed and it's pushed to the bottom, and the mitochondria plus rough endoplasmic reticulum, everything is more at the bottom and not at the surface. The other common feature is the myoepithelial cell. You can see them here. Yes, as we saw in the previous example, they are more of a star-shaped structure sitting between the cells and the basement membrane. Let us look at this bigger duct in detail. So you can see that, like most of the ducts, it's also having cuboidal cells with a round central nucleus, and all the cells are sitting inside a basement membrane. With further magnification, you can also see some of these myoepithelial cells, because even for the ducts, we need some compression from the exterior to push the saliva for easy movement. Here is a very good example of intralobular duct, otherwise called the striated ducts. So if I keep zooming in, see what has happened. We have the cuboidal cells and we have got the basement membrane. But if I isolate and zoom in the cell, you can see that the basal surface of the cell is highly folded. Okay, and this is the one giving the appearance like striations for the cell, and that's the reason these ducts are called striated cells. And what are those striations doing? They are nothing but infoldings to increase the surface area. Therefore, you can lodge heaps and heaps of mitochondria inside. So you can see that all the mitochondria are lodged inside and thereby increasing the metabolic activity of the ducts. So that is the advantage of having these striations. You can also see some of the myoepithelial cells here, like we saw in the bigger ducts. So we have seen one of the serous acinus in detail. We saw the basement membrane, we saw the pyramidal cells, organelles, and how the lumen is smaller for function. We also saw the mucus acinus with all its details and a smaller striated duct plus a larger metabolically inactive duct. We also have blood vessels inside. Here is a capillary. If you see a capillary has got a flat endothelial cell. Okay, so that is a kind of squamous epithelium, and you can see connective tissue keeping the endothelial cells in place. There is nothing much within the capillary. It is just flat cell, nucleus inside, and some connective tissue on the outside. You can also see small arteries. If you look at the arteries are yes lined with flat squamous endothelial cells with a flat nucleus, and you can see the connective tissue fibers on the outside, plus you can see lots of smooth muscles. This is how the artery is differentiated from a duct. So you can see that the duct almost always has a cuboidal epithelium, whereas any blood vessel, be it capillary, vein or artery, has got an endothelium which is nothing but a flat squamous epithelium. The other structure which is very typical of the submandibular gland is the mixed acinus. See here, you can see it is almost a complete mucous acinus plus a cap of half serous acinus, and this is what is called a serous demilune. You can see that they are combined, so that is why they are considered as unique acinus by themselves. Otherwise, all the other details are same. If you go into the mucus, you will see mucus droplets plus the location of the nuclei and the organelles, etc. Same with the serous.